Welcome to the launch of the Institute for Molecular Bioscience Centre for Superbug Solutions. It's a pretty wordy title, but what this centre will do is groundbreaking. It will focus on diagnosing infection and targeting the right drug to treat it. It will track infection. And perhaps the most exciting of all, it has begun the search for a new antibiotic that might be the answer to end superbugs. Just a figure that got my attention. By 2050, 10 million people will die every year because of microbial resistance. That will be more than the number of cancer patients worldwide. That's a startling reality, as you'll hear a bit later, and a reason why the IMB Centre for Superbug Solutions is just so important. My name is Madonna King. I'm your MC for the evening, and if there's any way I can make the evening more comfortable, please let me know. Before we start anything though, could I acknowledge the traditional owners of the land on which we meet and pay my respect to elders past and present and emerging community leaders. Could I also acknowledge Professor Peter Hoy, the Vice-Chancellor and President at the University of Queensland, Deputy Vice-Chancellor and Vice President Research, Professor Robyn Ward, UQ Chancellor John Story, and Professor Brandon Wainwright, IMB Director. Dr. Jeannette Young, the state's Chief Health Officer, Professor Jeffrey Lipman from the Royal Brisbane and Women's Hospital, Professor Matt Cooper, the Director of the IMB Centre for Superbug Solutions, and Matthew Ames, who is not only a bacterial infection survivor and my friend, but an incredible advocate in this space. Welcome to you all. A couple of housekeeping matters before we get underway. The restrooms are out those doors at the end of the corridor on the right-hand side, just past the staircase. Could I ask you to turn your mobiles to silent now, as we are videotaping this event and we're actually live streaming it. So I'd like to say hello to all of those watching us in both New South Wales and Western Australia this evening. And in the unlikely event of an emergency, if we could just follow closely the instructions given to us by staff, emergency exits are at the rear of the room. We encourage you to join tonight's conversation on Twitter. The hashtag is #Superbugs. Who's on Twitter? Good on you, so let's start tweeting. And now it's my pleasure to introduce you to Professor Robyn Ward, the university's Deputy Vice-Chancellor of Research, who will provide us with an overview of UQ's involvement in the fight against superbugs. Please welcome Professor Ward. So apologies for my footwear. I've got a broken foot and I do normally wear proper shoes, so excuse the sandals. So first off, I'd like to acknowledge the traditional owners of the land on which we gather and meet and pay our respects to elders past and present. To our distinguished guests, Jeannette Young, Professor Jeffrey Lipman, and to Matthew Ames, Mr. John Story, the Chancellor of University of Queensland, Professor Peter Hoy, the Vice-Chancellor, Brandon Wainwright, the Director of IMB, UQ colleagues, distinguished guests, ladies and gentlemen. Thank you, Madonna, for the introduction, and good evening to everyone. It's my great pleasure on behalf of UQ to officially welcome you to the launch of the new Centre for Superbug Solutions and COAD, the Community for Open Antimicrobial Drug Discovery. I came to Brisbane actually only earlier this year, and in fact one of the last patients I treated as an oncologist in Sydney died just before I left there. She didn't die of cancer, she died of a superbug. And I'll just tell you a little bit of her story. She was a woman in her early 50s who came to see me unable to swallow even her own saliva because she had a cancer at the bottom of her esophagus. And we treated her with chemotherapy and radiotherapy, and she was doing very well, although she was very malnourished. And we discharged her shortly after completion of her treatment, and she spent a little bit of time in a rehab unit near the hospital, and then started to develop some palpitations and came back into hospital, and within minutes of arriving on the ward had a cardiac arrest. She was resuscitated and then within about another 10 minutes also arrested again. And as is often the case in public hospitals, there weren't any beds to monitor her heart through the night, and so
we had, as a measure of last resort, to take her to intensive care just to have monitoring done. And unfortunately, she continued to have multiple arrests through the next few days. And then on the third night, she had a urinary catheter put in place. And unfortunately, the next morning when I came in, although I'd said, don't catheterise this woman, she unfortunately was catheterised and her urine was colonised by VRE. And very soon after that, she developed a high fever and multiple attempts to try and control her infection with now VRE septicemia were failed over a period of a number of weeks. We used old antibiotics, we used new antibiotics, and none worked. And after 6 weeks in intensive care, she passed away. And I think for trying to explain that story to her family was one of the more difficult things I have to do as an oncologist, in that they couldn't understand why their mother had been ostensibly had such a good result from treatment of her esophageal cancer, and yet that was dying of an infection that we had no antibiotics to treat. So I think you can imagine that as hard as that is to get your head around, how much harder it must be when people go into hospital for what ostensibly is very simple procedures, only to leave with all sorts of morbidities or even to die as a consequence of being infected with what would seem to be innocuous bugs, but for which we have no recommended or available antibiotics. So worldwide, the inappropriate use of antibiotics is responsible for germs becoming resistant to antibiotics. It's essentially too much of a good thing. Australia can't protect its borders from these germs. Germs don't need to travel to Australia in leaking boats. Many of them ride in the pointy end of a plane, colonising medical tourists coming back from Southeast Asia with a new face, large breasts, bum implants and a glowing smile. Each week in Australia, more than 170 people die from bacterial infections. Over 175,000 cases of healthcare acquired infections are treated in Australian hospitals annually, and the cost to the Australian economy is about $1 billion annually. So what's UQ doing about this problem? UQ's Professor Matt Cooper has led the development of the IMB Centre for Superbug Solutions. This has drawn together Australians' leading medical researchers, clinicians, pharmacologists, veterinary experts, drug developers, scientists and industry leaders. The centre is developing new diagnostics so that superbugs can be detected quickly. They are developing an online portal and a dashboard to allow better tracking of infection and provide government and healthcare workers real-time data on infections, resistance and antibiotic use. They are trying to fast-track the development of new drugs against superbugs. One such example is a potential new class of antibiotics inspired by sugar molecules produced by bacteria. This work has been done by Professor Cooper and Dr. Johan Zook, in partnership with the Queensland biotechnology company Alchema and other international partners. To help researchers understand how antibiotics work and what type of compounds we should be looking for in the future, the Wellcome Trust and UQ have funded the establishment of the Community for Open Antimicrobial Drug Discovery, COAD. This unique multi-million-dollar global initiative is screening compounds from academic research groups from anywhere in the world for free, with the aim of discovering a new class of antibiotics. So let me conclude now by just mentioning two other researchers at UQ who are also doing great things in this space. Dr. Nouri Ben Zakour from our School of Chemistry and Molecular Biosciences has tracked a multi-drug resistant E. coli strain, which is only one gene away from becoming resistant to almost all antibiotics. Ben can now try to develop tests to rapidly detect and help combat the spread of this germ. The second researcher is Dr. Hassoum Serwawi, a PhD student from the UQ Centre of Clinical Research. Hassoum won one of only 5 international Rolex laureates last year for his work towards creating a diagnostic tool to rapidly identify superbugs. Hassoum seems a fitting place to end this address, and he's one of the stars of UQ's new campaign to create change. And creating positive change in the world is at the heart of the IMB Centre for Superbug Solutions. So once again, it's a great pleasure to welcome you to this evening, and I look forward to the presentations and discussions. Thanks very much.
Thanks, Professor Ward. We can give all the statistics we like, but a story like that you told us kind of touches the heart as well as the head, doesn't it? It's now my pleasure to introduce Professor Dame Sally Davies, the Chief Medical Officer in the United Kingdom. Dame Sally is notable for many reasons, including her practical, common sense approach to important health issues. In terms of AMR, her pivotal 2011 annual report highlighted the importance of it to society alongside terrorism. Dame Sally Davies is unable to be with us this evening, but has recorded this important message for us via video.
Good evening, Madonna. Hello, everyone. I'm delighted to be with you all this evening to celebrate the launch of the IMB Centre for Superbug Solutions at the University of Queensland. So let me start by introducing myself. I'm the Chief Medical Officer for England and the Senior Independent Medical Advisor to the UK Government. So ministers seek my advice on all issues to do with health. And I also have a duty every year to produce an annual report, independent, on the state of the nation's health. And in order to fulfill this duty, I've taken an approach of splitting my annual report into 2 volumes. One which is about surveillance, and the other which is much more about advocacy. And my first advocacy volume focused on infections. I actually thought infections were going to be really safe, but that they would take me from the community into the hospital and cover the whole area. And I did not expect at that time to come across an issue that would then become the priority for my time as Chief Medical Officer. Now, as a haematologist, I was well aware of antimicrobial resistance, AMR, and we doctors and nurses and experts have known about that rise of antimicrobial resistance. So it wasn't that I found something new, but what I discovered was that since I stopped seeing patients, the inexorable rise has happened, and that actually there wasn't much action. And what I set out to do was give this issue and those experts a voice, not only here in the UK, but actually internationally. And actually, the first main recommendation in that annual report, published in March 2013, was from me to government that AMR should be put on the government's risk register. And, you know, it is that one recommendation that has led us to where we are now. Once something's on the government's risk register, at least in this country, action has to be taken across the whole of government to ensure we're doing everything we can to address this problem. So that means not only the Department of Health, but we all know this is a One Health issue. So the Department for Agriculture and Rural Affairs, DEFRA. This is a global issue. So our aid department, DFID, comes into it. And of course, if we're going to get real action globally, then our Foreign Office, Foreign Ministry, is very important too. But it becomes important for the whole of government, and it is a challenge that our Prime Minister has taken up personally. And being on the government's risk register, this is also non-party political. This is for the nation. As a result of all this strong support, and it is amazing how everyone has come together about this, we have now a really strong cross-government strategy for AMR with metrics and reporting, both on those metrics and already this summer, our first One Health report. We've led with Sweden and the Netherlands a strong resolution, a global resolution on AMR at the World Health Assembly last May, meaning that every country has now committed to develop a national action plan. And meetings like this all around the world, discussing what needs to be done to tackle AMR and how to take action, are part of that. Our Prime Minister, Prime Minister Cameron, set up and launched an independent review led by Lord Jim O'Neill to look at global solutions to stimulate the R&D pipeline, because not only is AMR rising, we haven't had any new classes brought to patient use since the late 80s for antibiotics. Other things we've done is last year the Prime Minister relaunched the Longitude Prize, 300 years after it was won for finding a way to say where you were on longitude. And the public voted through television for what they wanted as the issue. And I'm delighted to tell you they voted that what we really need most as the Longitude Prize issue is a rapid diagnostic to solve antimicrobial resistance. So that competition is running now. Our new battle to move forward is to get a whole day at the UN General Assembly in September 2016. We must not rest until we know that our children and their children will have effective antibiotics. This is a global issue. Take Europe. It is calculated that at least 25,000 people every year die of sepsis where the bacteria are resistant to infection. A similar number die in the United States every year. Now in Europe, that's the same number as road traffic accidents, and the cost is dramatic. In Southeast Asia, as you probably know, one child under the age of 5 dies every 5 minutes. This is catastrophic. And there are places in southern Europe where the bone marrow transplant units are regularly closed for deep cleaning because of superbugs, because of antibiotic resistance. Globally, we're seeing a dramatic rise of multi-drug resistant infections, these superbugs everywhere, resistant to standard and even last-ditch antimicrobials, and we've got nothing to treat them. Gonorrhea is becoming a clinical problem again. This is very worrying. And you know, antibiotics are less than a century old, but they've transformed society through improving health. Before antibiotics, 43% of deaths were caused by infections. Now it's about 7%. Interestingly, Alexander Fleming, when he discovered penicillin and then went to collect his Nobel Prize, prophesied that we would suffer from resistance and that this would be a big problem. And then we had the golden age. After penicillin, we had sulfonamides, but we had many other antibiotics. Classes discovered one after another, so that we arrived where we are with modern medicine based on antibiotics. And they have revolutionized life and medicine. Just think what underpins modern medicine. Cancer chemotherapy, not possible without antibiotics. Transplantation would be lost. A lot of routine surgery, including the caesarean section I had many years ago, the hip replacement I expect to have one day, underpinned by antibiotics. But this global age of discovery of antibiotics seemed to finish in 1987. And we have got a market failure. We're not incentivizing the production of new antibiotics. So we face the question, where are we going to be if we don't do something about it in 20, 30, 50 years' time? Are we going to be back with 43% of us dying from infection without modern medicine? A dark ages. And it's difficult because it isn't just one solution. It would be easy if it was. We've got to tackle this through a lot of different measures. And a lot of those are behavioural. We've got to get hand hygiene and basic hygiene and sanitation right. And actually in Australia and the UK that shouldn't be difficult. But this is a worldwide problem, and it can, as I'm going to show you in a minute, be difficult. We can lose it. We've got to, as I've said before, incentivize the pharmaceutical industry to produce new antibiotics or find different ways of doing this. And actually, the science is really difficult. So it's not just reimbursement, it's also the science that's difficult. I've mentioned rapid diagnostics. Clearly, we can conserve antibiotics if we have rapid diagnostics. We've got to look at global prescribing practices. Our National Institute for Clinical Excellence has just launched guidelines this August about prescribing in general practice. And they reckon that 22% of prescriptions could be saved if we've abided by the best guidelines. And what about patient behaviour? Those patients who feel they're validated, they're truly sick if they get an antibiotic when it was a virus infection that won't respond. How do we stop them wanting these wonder drugs? How do we help them to understand antibiotics are a public good? We've got, all of us as doctors, to prescribe when clinically relevant, and that's just the human element. Think about the widespread use of antibiotics in agriculture and animal farming, aquaculture, fish farming, and how they are abused around the globe. You've got some slides on your seats, and if you look at them, you can see how, through concerted action in England, we've made a difference to infection, to MRSA, Clostridium difficile. We needed, first of all, to re-find good hand hygiene in our hospitals and communities, and to take action. And if we don't take action, as you can see in those slides, then what we're going to see is the end of modern medicine. I've talked about it, losing cancer treatments, losing surgery. We will start to see people dying from something like a nick from shaving, a cut from gardening. So programs such as Community for Open Antimicrobial Drug Discovery, which you're engaged in, could well hold the key to antimicrobial drug discovery in the future. This is really important work, and I welcome the role that you're playing in our global fight against AMR. This is truly a global fight, and we all have a role to play. I want to thank you for coming to the event, and I hope you're going to have a really lively panel discussion. I'm so sorry not to be there in person. I wish I were, because I know I would learn lots, and the energy that you put into it is important to me on the global battlefield around AMR. But I've got a question to get you all started. What is Australia doing in the AMR space to fight superbugs? Thank you.
An excellent question, isn't it? And we do thank Dame Sally for her time, and we'll address that question in our Q&A. And Dr. Jeannette Young, I can see that question coming straight for you. And now to Matthew Ames, who I would like to welcome up here. Matthew's story gripped our nation, but it also should be the impetus for action. Matthew was 39 when what started as a sore throat led to the loss of all 4 of his limbs. He'd contracted streptococcal, resulting in toxic shock. No one thought he would survive. But driven by 4 young children and a wife whose determination beats his, I suspect, he is not only here to tell his story, he spends much of his time now helping others and urging our policymakers to take disadvantage more seriously. Ladies and gentlemen, please provide a warm welcome for Matthew Ames.
Thank you, Madonna. Distinguished guests, ladies and gentlemen, tonight I want to take you back to about just over 3 years ago, as I was lying in a hospital bed with my wife beside me, describing to me that while I'd been in a coma for 3 weeks, I'd had all of my arms and limbs removed. Once the initial shock of that particular news passed, I started to ask some questions. One of the key questions is, why did this happen? I now know that the simple answer is bugs. That is bacteria. When I woke up, I remembered how it all started. I remember I had a sore throat that deteriorated starting on a Friday night, deteriorated over the weekend, and then on Sunday night I thought, something's just not right. I knew there was something wrong with me. So I went into the emergency department, being a Sunday night, and I was sent home with the diagnosis that it was the flu. At that particular stage, I had a small rash on my left forearm, and that got larger over a couple of days, and a number of other things continued to deteriorate in my health. So I sought additional assistance, and I saw some doctors on the Tuesday night and the Wednesday morning, again with the view that it was the flu. So by Wednesday night, I could no longer walk unassisted. So I took myself again to the emergency department. I remember sitting in front of the triage nurse explaining my symptoms, and that's the last thing that I can remember. So Diane, my wife, then had to fill me in on what happened after that point. My body went into septic shock that Wednesday night, and then I was put on IV antibiotics, and the following morning the doctors operated on my left arm, which was where my rash was, in an effort to stem the infection. Unfortunately for me, that course of action didn't quite work, and by the Friday evening a decision was made to remove my left arm, which was thought to be the main source of infection. 24 hours later, things still hadn't improved, and they'd actually got worse. So on a Saturday, my wife had to make the heart-wrenching decision to either let me die or to remove the remaining 3 limbs that I had left at that stage for a 1% chance of survival. I am one of the lucky ones. There are many who don't make it. It's easy to listen to the statistics, the numbers, the probabilities, but I am one of those numbers. Any one of us, or those that we love, can be one of those numbers at any time. I do wonder, how did a fit 39-year-old man with hardly any medical history to note end up in this position? So when Matt Cooper from the centre came to talk to me and said, would I be involved, I was more than happy to help. I don't want anyone else or their families or their friends to go through what I've had to go through. I have found it somewhat difficult over the past few years, but the journey, knowing that I can actually help out, has been a fantastic one. For me, the things that this centre is doing is particularly critical. When I think about awareness and education, if I'd had the right antibiotics at the right time, the chances are I would have my arms and legs today. If diagnostics had been available so that I could get early treatment and rapid treatment, it's highly likely that I'd have my arms and legs here today. And if there were more variety of antibiotics available, there's a high chance that there may be something there that would have helped my body fight off the infection. My injuries may have been less, or the side effects that I suffered from the strong antibiotics may have also been significantly diminished. So my hope is simple, that we all get actively involved in making this centre achieve some amazing things. Not because it's the right thing to do, not just because it's the right thing to do, not just because of the numbers and the statistics. Because my hope is that all of us, all of you, everyone around the world can continue to enjoy the simple things in life that we take for granted. To play music, to kick a ball with your kids, to walk your daughter down the aisle, to hug somebody, and sometimes simply to hold hands. We can do this. It just needs you. Thank you.
Thank you, Matthew. You know, pushing the boundaries is what Matthew does so well. And in my local neighbourhood, you see it every day in Matthew picking up his children and walking them home with them hanging off his chair, or he's volunteering to be a parent helper on school excursions, or his ability to use what's happened to him to make it so much easier for everybody else. Please put your hands together again for Matthew Ames. Now, every launch needs a bit of celebrity, and Matthew, that's not you, nor is it me. I'm going to introduce you now to TV host, writer, and former Bond girl, Daphne Kershaw, who has a very important message.
Hi, I'm Daphne Kershaw, and I want to share my story with you. Last year, I started to feel ill, and the doctor thought I had a bladder infection. She gave me antibiotics. I finished the treatment, but the pain and the tiredness never left. So after a while, I went back to the doctor and got some more antibiotics. And I ignored the fact that even after taking antibiotics, my symptoms never really went away. I just thought I must be tired, I need a vacation, maybe it's all in my head. Because after you take antibiotics, you're cured, right? This is what we have come to believe. Antibiotics are strong. They cure all, and they make you better, until they don't. And then one evening in September, just after I had hosted a live television show, I just fell apart. I started shaking violently, and I ended up in the hospital on an IV drip for 5 days. Well, after that, I thought, oh, I'm healthy again. But no, the infection came back, and again, and on Christmas Day, only 6 months ago, I was back in the hospital. And this was the first time the urologist said to me, your bacteria is multi-drug resistant. She said, for this type of E. coli, we can use 8 different kinds of antibiotics. But in your case, 7 of those do not work anymore. So now I'm taking number 8, and it works so far. But what will happen if this bacteria learns to defend itself against number 8 and will become resistant too? I don't know. Nobody knows. What I do know is that having such a bacteria and a urinary tract infection and infected kidneys is very, very tiring. I have never really experienced the type of impact a bacteria can have on your life up until now. When I have to make an appointment at the hospital to see a neurologist, she says to me she has to see me at the end of her shifts. Why? Because after my visit, she has to clean and disinfect everything. I was quite embarrassed when she told me that. Well, she didn't mean to hurt my feelings, but it made me realize I am a patient with a multi-resistant bacteria, and this is not something to take lightly. It's a serious thing. They even said to me that if I ended up in the hospital now, they would have to put me in quarantine. In quarantine? I'm a mother of 2 children, so of course I'm wondering, am I contagious? This whole situation was starting to make me a little bit nervous. I thought, how did I even get this thing? I know a lot of my friends do not really believe me when I tell them that this multi-resistant bacteria are very real threats. The most common reaction I get is, so if these antibiotics aren't going to work anymore, what is the hospital going to give you then? Well, how about nothing? There is no alternative yet. I'm not a doctor, I'm not a scientist, and I'm not a politician. So I do not know what the solution to this problem is. But I do know, as a very surprised patient who ended up with one of these superbugs that I can hardly get rid of, that action is needed. My urologist told me that more and more young children come into the hospital with bacterial infections that turn out to be multi-drug resistant. These kids are like 7 years old and already resistant to 5 or 6 different types of antibiotics. But these children have never had an antibiotic treatment in their life. So how did the bacteria get resistant? This must mean it is everywhere. You breathe it, you swallow it, you eat it, you drink it. But this has to stop. And that will take all of you, because it's a group effort. It's a whole field of problems that we are facing. My grandfather was a farmer, and he used to say that to attack a whole field of problems, we should adapt the behavior of cows by grazing, dealing with each patch of the problem at a time and clearing up one area before moving on to the next. With a huge amount of antibiotics going around, it is time to start asking ourselves, are we on the right track here? We should not be defeated by bacteria, and we should definitely not be defeated by our own passive attitude. So tonight, I encourage you to join this discussion and help us fight superbugs.
Yeah. As Daphne says, she's not a doctor, a scientist, or an academic, but she sure can tell a story, can't she? Can I just ask how many people in this room have ever taken antibiotics? Just raise your hand. Sometime. Everyone. Now, if I walked down Queen Street Mall and asked that same question, it would be a similar response. And that is why the work of our next guest and his team is so vital. Professor Matt Cooper is the director of the IMB Centre for Superbug Solutions. And Professor Cooper has an international reputation in this area. He's known as a scientific entrepreneur with a track record in both innovation and in leadership. He's driven multidisciplinary programs in electronics, in physics, materials science, chemistry, microbiology, inflammation, and diagnostics. His R&D teams have launched many product families to market and now have a focus on drug discovery for new antibiotics, diagnostic platforms, and breakthrough treatments for inflammatory diseases like Parkinson's, like asthma, Alzheimer's, diabetes, arthritis, gout, and cancer. Ladies and gentlemen, please welcome Professor Matthew Cooper.
Ladies and gentlemen, distinguished guests, thank you so much for coming. We really appreciate it tonight. And Matt, thank you so much again. I should point out that Matt is part of the team. He comes to our operational meetings. He's an incredibly talented individual, amazing project management engineering experience. And so, Matt, I'd like to thank you personally for coming tonight. So we've heard some pretty inspiring and dramatic stories, and there are a number of slides which we could talk about in terms of statistics. If we could do the next slide. Or not. I'll just give you a wave there. And we've heard a lot about this. I want to give it back to some personal stories. My father, John Cooper, is in the audience, and he was born a long time ago. And he lived up in Lura in the Blue Mountains, and they were a large family. I think it was 10 brothers and sisters. And one of his brothers died at the age of 4 or 5. He had a pneumonia. My father told me he remembers his brother, very young, having a chest cough, was taken away in a car. The doctor in Katoomba was a bit of a way. Never seen again. And at that stage, it was, during the war, World War II, there were no antibiotics. So we forget exactly how valuable these compounds are. In science and medicine, we talk about cures, a cure for Alzheimer's, a cure for Parkinson's, a cure for cancer. Well, it's an overused and ill-advised word. There are very, very few medicines that cure disease. But in this case, they are miracle drugs. We've forgotten that. We've forgotten that we can cure infection. Before antibiotics, as you heard, 40% of people died from infectious disease. We've treated them as commodities. We expect to pay $20 to $40 for a course of antibiotics. We are paying $50,000 for cancer medicines, and that's a great thing to stop cancer. But the other side of the story, I was fortunate enough to go to Cambridge University, and my professor was an FRS and 400 papers and a very, very bright man. He had a hepatocellular carcinoma, a liver cancer. Fortunately caught early, 95% plus survival rates. He went to hospital, his liver was sectioned, they cut a little bit of his liver off. He picked up a drug-resistant bacteria and died in 4 days, having spent his whole life working on antibiotic research. So once again, we're going back to the future. And these are the stats here. Next slide, please. This is what's happening. These are Australian Bureau of Statistics numbers of what will happen if we do nothing. And many people have said the greatest sin is inaction. It's very easy to watch this happening. As with climate change, as Dame Sally said, this is a complex issue. It involves animals, economics, policy, research, and we need to break that down and attack it. Next slide, please. So what we're doing is we're trying to take a unified approach. This won't be solved by chemistry or biology or government policy, but combining this all together and putting together clinicians, chemistry, microbiologists, pharmacology, all in one centre is a good step forward. It will engage egalitarian debate. We may get some new innovative approaches, and I'd love to tell you about a couple of those today. Next slide. So we have taken 3 approaches to this. The first of these is the diagnostic you heard about, and I'll tell you a bit more about that in the next slide. But fundamentally, as Matt told you, as with cancer as well, early diagnostics and early prognosis enables survival. If you give the right drug in time, on time, we can save lives. It's a very, very simple thing, but funnily enough, we haven't done this in this field for quite a long time. The second activity of the centre is in understanding the problem. We all have limited resources. The health budget is limited, the federal health budget is limited, but if we can better allocate resources to where the problem is and highlight those particular hospitals in particular where we need extra resource, it's going to be a win-win. We'll save money and we'll save lives. And then finally, I'll tell you a little bit about the search for new drugs. One part of the solution, of course, as you heard from Daphne Decker, is we're going to need new antibiotics. And it's getting harder and harder to find those, and I'll tell you why. Next slide, please. So if we go back again to diagnostics, there are 2 gentlemen on the slides here, Louis Pasteur, and he's the father of microbiology. He was the first scientist, the Pasteur Institute, pasteurization of milk we're all familiar with. He showed that bacteria were the cause of infections, and bacteria are microorganisms. Before this seminal discovery, we thought of diseases such as cholera as ethereal diseases. They were this transpiration through the air. John Snow, of course, the father of epidemiology in London, showed that these diseases were caused by transmission of microbes. And the other gentleman on the right, who's a fellow of the Royal Society, is Dutch from his name. He's a cloth merchant. He's actually the inventor of the compound microscope. So he wanted to look at the waft and the weave of his fabrics to determine whether or not his suppliers from the Dutch East Indies were giving him the right stuff. Very, very bright man. He invented the compound microscope so he could look at fabrics more carefully. And he discovered this amazing universe of microorganisms. You know, he found this weird, wonderful, microscopic universe of animals. And he wrote a letter to the Royal Society and said, look at this. And they said, you're Dutch. This is crazy. Go away. But he had perseverance. It's one of those great maxims. What do you need in life? You need motivation. What gets you out of bed in the morning? What inspires you to do what you do? You need some perspiration and hard work, but the key thing is perseverance. Keep on going. So he kept on writing to them and eventually was awarded that. The reason I show these 2 gentlemen on the next slide is that if you look at modern microbiology, almost 300 years later, we haven't changed that much. Those two gentlemen could come into a lab and say, okay, I can recognise this, 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 and this. After 300 years of technology and science where everything else has changed phenomenally. Communications, the way we manage diabetes, it's all changed. But we basically take a sample from a patient, we culture it, which takes a minimum of 12 hours. We then look at it, we put it on a plate, we culture it again. You'll hear from Jeff Lipman later on. It can take 48 hours before we can say, okay, we think it's bug X. And for patients such as Matt, that's a long time. Sepsis survival is measured in hours, not days. And it's unacceptable that we have such a recalcitrant approach to diagnostics. We can do better. And the reason I'm here today is in terms of impact of what we can do. A lot of these deaths and suffering is preventable. I hope we find a cure for cancer. It's really hard. But I know that we can do this. Many of these deaths and many of these things that are happening, we can fix now. We just need to work together. Next slide, please. So on the diagnostic front, we've taken an approach that involves, as I said, informatics, genomics, chemistry, putting it together. And on the left, what you'll see is we start with a sample. And the first thing we need to do is get rid of that culture step. We can't afford to wait 24 hours or 48 hours because those patients could be dead in 24 hours. So we take a sample of 10 mils of blood or urine and we pre-concentrate the bacteria from 10 mils down to a small volume. So we've actually magnified that signal. And then we use new technology, which is not our technology, it's from the UK. And we look at the nucleic acids or the DNA in that sample. So this is what we call a whole genome sequencer. And Fred Sanger, who has 2 Nobel Prizes from Cambridge again, first invented Sanger seating, and the first genome cost $3 billion to sequence for human genomics. We can now sequence a human genome in about 4 days, cost about $3,000, $2,000. It's a big machine. Well, this is a sequencer you can put in your pocket. It plugs into a USB stick with our front-end technology, together with Lachlan Coyne's group. We'll build algorithms that can say, okay, within 10 minutes after this first step and the pre-concentration, we can say it's bug A, it's Klebsiella pneumonia. Another 10 minutes later, we could say, okay, we think it has these particular strain type characteristics. And then about 4 to 6 hours later, we can say it has these particular resistance types. That result can then get emailed to the clinician. I've got an app on my telephone I can show you later. It could be a paradigm change. We've got to get speed and technology into microbial diagnostics. It's not acceptable that Louis Pasteur and Anton van Leeuwenhoek can still recognise many of our technologies. Next slide. The other approach is more complex, and this is where governments have to lead. I would say in many aspects the UK, and you heard Dame Sally, she's a phenomenal woman. She's made more difference to UK policy engagement. Obama's just launched a $1.2 billion fund in the US. Cameron's behind with a £900 million pound fund in the US and the UK. The European Union put 900 million euros behind this. Australia, unfortunately, spends 0.6% of its medical research budget on this issue. So we're about a decade behind. But in terms of that minute of resource, we can actually do better. We can look at hospitals and say, okay, which hospitals are performing well? Which hospitals have more infections or less infections? We can also mine Google Trends and other databases and say, okay, what is the community worried about? Are they searching for flu? Are they searching for superbugs? Understand in a holistic framework where the problem is so we can allocate resource. Next slide, please. And the last thing, of course, is the next drug. You heard from Dame Sally once again. The last real breakthrough in this field was in the 80s. Unfortunately, pharmaceutical companies have left the field. There's a very simple reason behind that. It's money. So pharmaceutical companies are for-profit companies. They develop money. They spend a lot of time and risk on clinical trials. And they've decided that the economic framework for antibiotic sales is broken. What we call the net present value for antibiotic development now is almost zero. And that's because we don't value the drugs. Again, we expect to pay $100 for a course of antibiotics, tens of thousands of dollars for other medicines which we take chronically. In a way, we're kind of stuck because antibiotics are wonderful drugs. They often work in 2 weeks and you never take them again. You forget about it. But the other reason, of course, is that this is scientifically very hard. Scientists now are going into caves, they're going into, the Antarctic muds and ices. We're looking in all these weird places to try and find the last antibiotic we could discover. In roughly 80 years, we've squandered this this incredible resource of compounds. But there's another approach, and this is kind of crowdsourcing. So we're familiar with the World Bank, which is a bank of money. Sperm banks are a bank of sperm. Tissue banks, seed banks in Norway. We've got these collective banks which humanity puts together. But no one's thought of doing a molecule bank. And all drugs are molecules. They're small chemistry molecules. We know that as chemists around the world, we make millions of compounds. So we thought, okay, individually these compounds are worthless. But collectively, if we can put them together as a resource, as a community, they're incredibly valuable. So the Community for Open Antimicrobial Drug Discovery, which is funded by the Board of Governors of the Wellcome Trust and the University of Queensland, is doing exactly that. We're reaching out to people all around the world to help us discover the new drug by sending in compounds from chemistry departments around the world, and we will screen them for free. We don't make any money out of it. It's not about IP. This is purely for public good as a community service. Next slide. And what you used to do, of course, in discovery was just pick up some dirt, separate the compound, and look to see if it killed the bug. And we're kind of doing the same. Again, we're going back to the future, but this time doing it on a global scale. We couldn't do before email, the internet, and all the things we have today. Next slide, please. And this is just basically one of the part of the issues. If we look on the right-hand side, that's all these grey dots are drugs. This is chemical space, the chemical universe, if you like. On the right-hand side is where the pharma companies have been looking at in the green circle. And the red dots are antibiotics and where they are in this chemical space. Imagine looking at the stars. So if you think about that, we've kind of been looking in the wrong place. It's another reason why we're not finding new antibiotics against the superbugs. So final slide. Second final slide. And I'm very happy to report the community response has been amazing. We're looking at agreements with the government of India, the National Collection of France, US, South America. This really is a global activity, and I'm very proud that it's based here at the University of Queensland and in Brisbane. We're the first group to sort of say, let's work together globally and bring these molecules together. Next slide. Early days, we've launched this in February. It takes a long time to get a new drug to market and discover things, but if we look at the success rates, these are two different types of bacteria on the left and the right. And if we screen a commercial collection, that pharma collection I showed you before, success rates are 0.008%. It's really hard to find. With the co-ed approach, we're getting much higher hit rates. So it's early days, but very encouraging that we're getting new chemical diversity. And final slide. And look, finally, it's my privilege to be up here today, but we have an amazing team. They come in at 4 o'clock in the morning. They come in on Sunday. I get emails at 1 o'clock in the morning. I can't speak highly enough of the team. There's 15 people alone in co-ed and probably 30 people in the centre. And we're all working as hard as we can. So I'd like to thank them from the bottom of my heart. And also I'd like to also thank Ruth Neal in particular, who's done a wonderful job organising tonight's event. So please join me in thanking the team.
Thanks, Professor. And we all want to hear more, don't we? So could I please ask you to remain on stage and invite Matthew Ames back up here? I'd also like to introduce you to two further panellists. The State's Chief Health Officer, Dr. Jeannette Young. And Dr. Young has a long list of responsibilities, from health disaster planning to aeromedical retrieval services, licensing of private hospitals, policy regarding research, organ and tissue donation, blood, poisons, medicines, cancer screening, communicable diseases, environmental health. The list goes on, but she's up here, so I'll stop there. It's wonderful that you're joining us, Dr. Young. Thank you. Thank you. And our final panellist is Professor Geoffrey Lipman, the Director of the Department of Intensive Care Medicine at the Royal Brisbane and Women's Hospital. And his research interests include all aspects of infection management in intensive care. And indeed, I think he's worked in intensive care units since 1979. So, Professor, thank you for your time tonight. Now, our panel is happy to take your questions. So if you've got one, I will come to you in a little while. Please just put your hand in the air and we will make sure we get a microphone to you. Or you can tweet your question, remember, to #Superbugs. So let me begin the conversation, though, with a question posed by Dame Sally Davies, and I think it was along the lines of what was Australia doing in the AMR space to fight superbugs? Dr. Jeannette Young.
Well, thank you. I probably shouldn't be speaking on behalf of Chris Bagley, who's Australia's Chief Medical Officer, but he's been working with Sally Davies and well aware of what the UK is doing. And here in Australia, we, of course, were one of the signatories to that WHO initiative that you heard Sally Davies speak about, where WHO is asking every country in the world to develop a strategy for antimicrobial resistance. So Australia has developed that strategy, and that was launched around 6 weeks or so ago, and we're now having a workshop bringing all the various players together from around the country in November to work through how we'll actually implement that strategy in Australia.
Are we moving fast enough, Professor Cooper?
I'd say now we are. It's been, it hasn't been a bipartisan issue. So where it made a big difference, as you heard with David Cameron, was the Prime Minister got behind it. Part of the issue is, part of this is with Treasury, it's with agriculture because of the use of antibiotics as growth promoters, it's in health. And in Australia, for various reasons, everyone, it's not my problem, that's health, it's not my problem, it's agriculture. But both Barack Obama, the EU, and David Cameron said, no, this is a serious issue. I'm going to control this and take care of it. So the document that Jeannette referred to is fantastic. It has recommendations, it's very well written, has lots of input. The key then is policy. Our politicians enact policy, and we need strong leadership to make this a bipartisan issue across all departments because it affects everybody. And
it comes to priorities, doesn't it? What's the priority of the day?
Yeah. Well, the argument for Treasury is very simple. It's bad days. If you go to a hospital and you pick up an infection, you're either there for a couple of weeks in the ICU or you're not there. That's not good for economics.
So you mentioned hospitals. Let me throw to Professor Lipman. And we've heard some stories tonight, but take us inside a hospital. How often are you seeing someone, a patient, suffering from antimicrobial resistance?
Far less than Europe, and far less than particularly Southern Europe. I think our biggest worry is the bringing in of these infections from overseas. I think when you go to India, and excuse me, I apologize if I offend anybody, but anything you eat and drink in India is going to colonize your bowel, and the problem then becomes, most of us when we go to India, we come back with some resistant organisms in our bowel. And that doesn't affect you today, but if you get infection, that can affect you. And if for any of you, excuse me again being a bit crude, if you go to India and you need a prostate biopsy, you've got a problem because your bowel is colonized with resistant organisms, and that biopsy has to go through the rectum, and you just don't want that.
So are you seeing it as a real problem now, or are you seeing it as a tsunami over the horizon?
A bit of both. Obviously at an individual patient level it's a problem. But from an Australian point of view, it's far less of a problem, and we're waiting for the tsunami to come. So it's both.
So Professor Cooper was talking to us about the time it takes to diagnose a patient and how that's done. Inside a hospital today, how is a superbug diagnosed, and how long from suspicion to evidence?
By and large, Matt's correct. The patient comes in, you see the patient, you hopefully diagnose an infection, which may have been a bit of a problem, but you diagnose an infection and then you start antibiotics soon after you diagnose the infection. And if you're in septic shock, the longer the delay to the correct antibiotic, the higher the morbidity and mortality. So what we do as a global phenomenon is to treat as many of the organisms as reasonable up front. Now you can't give 10 antibiotics and antifungal agents and antiviral agents to everybody with an infection. while you wait for the diagnosis. So you treat what you think is a reasonable chance of covering the organism, and you're right most of the time. But I think one of the problems, which is slightly different, is that antibiotics to this patient is the one set of drugs that affect other patients as well. So when you take a blood pressure tablet, you're treating that patient, and that patient's blood pressure drops. When you give that patient antibiotics, his organisms in his bowel change, and indirectly you affect the ecology of other patients as well. So although we treat this patient and we try and diagnose, we have to start broad, and then we often tailor after the 48 hours. But it's before you get the exact organism that you're already causing potential problems, and you're over-treating to a large extent.
So, Matthew, we could listen to you all night, but you actually caught a superbug in hospital. Is that right?
Yes, I did. I got an MRSA in some of my wounds from the hospital, which has made me a little bit balder in the back of my head a bit earlier than I thought I was hoping to. But yeah, it took a fair while to actually get rid of those as well, which was a bit of a complication afterwards.
And was that diagnosed quickly? That part of it? Was that diagnosed quickly?
It probably, with all the other things that were going on with me, wasn't the
priority.
The priority, but yeah, it took a few days. But MRSA is a pretty standard test, which takes a little while, but they did pick that up.
So we're talking about the frequency of this happening in hospitals. And Dr. Jeannette Young, just to pick up on something that Professor Cooper said, he talked about, at one stage being able to compare different hospitals. And I know this was on the horizon a couple of years ago. As a public health official, would you support a hospital comparison in superbugs?
We already do it in the public sector. We don't have that same degree of transparency with the private sector, but at a national level, the Commission of Quality and Safety is looking at that now. So we report our Staph aureus bacteremia rates, and they were actually being reported in all of the newspapers around Queensland every 3 months. They're now online, so rather than putting them in the media, they're there for people to see online. So you can see every single hospital and health services infection rates for one particular infection. So there's that's available now.
Where do others sit on that? Matthew Cooper, I mean, that would mean presumably that some hospitals that have the most dangerous cases are going to be higher up the order, aren't they?
Yeah, Jeanette's right, the data's there, but I guess the UK took it to another level. They made it visible, and they did it every week, mandatory reporting. They also set targets, and if a hospital was underperforming, resources and teams would go there to train people, even to the extent of the CEO of the hospital. The CEO's bonuses were tied to metrics, right? So some of the data is there, but I think we can do better. We would love to have some of this data visible. The first part of solving a problem is visibility of the problem.
And you say visible, it's online if you go searching for it. What kind of visibility do you want?
Oh, look, this is a matter for government, really, to be honest, and Jeanette's right, that the data is there. We think that now what we can do is combine these data. So we can combine Google Trends, what the public's looking for, put it into a portal. It could be the Director General or the Minister of Health has top-level access, and if you're worried about a Hendra outbreak or a flu outbreak, you can log in and see things. But I believe in open data. I think the more accessible it is, the more opportunity we have to change things. Professor
Lipman, you're at the coalface. Is that something that...
No, I was just thinking that I think that's part of it. The diagnostic part, which you asked me about, I think is going to revolutionise. There are two big issues in intensive care. One is the delayed diagnosis, right? And the quicker we can get to the right antibiotic at the right time, the less what we call collateral damage. So that's vital, and Matt and other groups are bringing point of care to us. But the next big diagnostic is to differentiate the viral infection from the bacterial infection, right? And which bacteria, is it the MRSA or is it the Klebsiella, because the treatment is different. So we're excited with this point of care technology, and I have no doubt that that type of technology is going to change the way we prescribe and indirectly cause less damage and preserve the antibiotics that we have today for future use as well.
Okay, that's probably a good segue to ask you, Matthew Cooper. The search for the next antibiotic is called the Community for Open Antimicrobial Drug Discovery. But you mentioned how it involves chemists around the world sending their compounds that they have lying around on their shelves here to Brisbane, to your unit, where you'll work on them. How is that going to happen? Has it happened, and where are you sourcing it from?
Everywhere. Anywhere we possibly can. In a way, we've been given by rules. We have rules of drug availability, bioavailability. You can't do this, you can't do that. And Peter Hoyle will know I don't like rules so much. But the fundamental thing is that we've got 100 million compounds that we've made, synthetic compounds that mankind has made. That's a lot of compounds. Every day we make 15,000 compounds. But of course, if that compound stays on someone's shelf, it's, I was a chemist, I did a PhD, I made 300 and something compounds. They end up in the bin. So that, think about diversity, think about a rainforest. We're kind of dominated by these monocultures of fields of wheat and potatoes, and we just grow this, we just grow that. We've already looked at those. We're not going to find new types of plants there. If we go to the rainforest, this amazing diversity of compounds, we're more likely to find an unusual, special plant.
Terrific way of explaining it. But so what does the antibiotic pipeline look like to you?
So clinically at the moment in phase 3 we're not too bad. The reason is that those compounds were discovered about 20 years ago. And so phase 3 is the last clinical trial phase before it goes on the market. You can buy it and the chemist will get it from Jeff. If we go earlier on, it's dire. We've actually put a survey out this week. It's called Antibiotic Researcher and Endangered Species. We think there are less than 1,000 people on the planet now working on this issue. There are more people writing white papers and recommendations and analyses than there are working on the issue. And for me, you're defined by what you do. And so there's a worry for me in terms of generations, and talking to my father and my kids are here, is that knowledge is driven by failure. Drugs fail, people know why they fail. There's no book on how to discover an antibiotic. And these people are retiring or getting fired, farmers leaving the field. So if we don't act in the next 10 years, the next generation will have no one to learn from.
Where and when are we likely to find the next antibiotic? Difficult question, but where are you thinking? And I'll go to Dr. Young and Professor Lipman on this too.
So it's a tough question. Well done. 2 things. One is diversity. Don't pretend you're smart. Sample as much diverse space as possible. Look places that no one's looked at before. To stand out, you first have to stand alone. The other one is going back to the future. Go back to the 50s and the 60s when it was the golden age. We found 12 antibiotics every year, right? So there were some dodgy ones that maybe had an issue with toxicity in the kidney or, caused a problem. So we're doing both in our group. We're going back to old ones that were forgotten. And they never went to the clinic, which means there's no resistance because they never got used. But then use your chemistry and your science to try and tweak them and make them less toxic. So we have 2 examples in our group where we've taken a very old compound from the 50s or the 60s and we're changing it to try and make it better. But there's only a limited number of those. Fundamentally, like everything, I believe if you have the right people and enough time and the funding, you can do anything. So I think chemical diversity is key.
What's enough time mean?
Well, when do you start? Do you wait another 10 years? It's like climate change. This is a global issue. You've heard from everybody. It's one of these few cases where how I behave, whether I take antibiotics, affects what you do in your health. It's a community issue. It's one of those issues where it's not just your disease, what you do and how you behave. If you go to the GP and ask for I've got an earache, I must have antibiotics, it may be viral. So let's start now.
Yeah.
Why wait?
So over to you, Matthew Ames, and you'd like to actually see more antibiotics on offer. You'd like to see more antibiotics on offer. Why is that?
Well, for me, the antibiotics that were used They'd started with a broad spectrum because they didn't really know what I had. Turned out that it was about the right one that I needed, but they were not winning the battle, so they upped the toxicity of some of the antibiotics that they use. So it was a bit of collateral damage. So that's why it's a bit hard for me to hear your questions, because I've lost pretty much all the hearing in one ear and half the hearing in the other. And that was a side effect of the antibiotics that they needed to use on me. So yeah, there are, even with the ones that we do have, when you get to that level, they can start to get a bit dangerous to use.
Professor Lipman, you're nodding. You want to say something there?
No, I'm nodding.
Yeah, agreeing. Okay.
I had a thought that I was... just move on.
All right. So one thing, and this is reflected in many of your questions sent to me already, and Dame Sally touched on it. She talked about hand hygiene. Dr. Young, do hand sanitisers stop antimicrobial resistance?
Well, they stop organisms.
Yeah, stop organisms.
So, yes. So we have a program in Queensland and throughout Australia, 5 Moments, which is the WHO-endorsed hand hygiene program that's regularly reported on, and you can compare different hospitals as to their hand hygiene rates. And it's a very important program.
In our homes, what's the difference between that and good old-fashioned soap and water?
Oh, soap and water is always superior. Oh yes, definitely. If you've got soap and water, you use soap and water. That's far better. But if you don't have soap and water, or you're moving very quickly and you're regularly washing your hands with soap and water and you just want to do an extra quick clean and you can't see your hands look perfectly clean, then you can use the alcohol gels, those gels. And they're a good thing to keep in your handbag and to use when you don't have access to soap and water. So they're better than nothing, but they're not as good as soap and water.
I have to add that we have, when visitors come to our laboratories, we ask them to put a handprint on a plate. We compared the Minister of Health, Cameron Dick's hands, and our Chief Health Officer's hands, and they were both pretty good.
Were they? Because I know I had mine done there too, and it's like waiting at a doctor's surgery for a result, and I hadn't heard after 2 days, so I phoned and said, oh, you know that hand thing? I do have very young children, and you know, there were a lot of door handles that I touched on the way in. I think mine was okay though. Probiotics, Professor Lipman, what role do they play here?
At this stage, certainly for intensive care, no role. For infant problems and infant diarrhea, they may have a role, but the data for intensive care is not adequate yet. We don't know exactly what probiotic. So when the bottom line is, when you take when I give you an antibiotic, I'll change your bowel flora. That's a given. That's why people get a bit of diarrhea. That's why we get thrush. You change your flora with the antibiotic that you give, and that's an effect of the antibiotic. So theoretically, if you're killing the organisms in your bowel, you want to give new organisms that will protect your bowel, in other words, probiotics. But we don't know exactly which bacteria, how much to give and how to give it. So for intensive care, probiotics is not good, is not valid yet. Yeah. The data then becomes a bit difficult to sift out whether you take yogurt or other things. I don't think, certainly for intensive care, it's not there.
Yeah, okay. I want to go to your questions in just a moment. So if you have a question, now is a good time to raise your hand and we'll get the microphone to you. There's one up the front. But before I do that, Matthew Ames, a mental resilience helped your survival, I suspect, too, and from the way you spoke. How important is that in this equation?
It's everything, really. You know, I think, I was lying there, going back to that time when my beautiful wife Diane was giving me that, that information. I just had this overwhelming sense that everything was going to be okay. Which seems like a crazy thing to kind of think in that moment, but it had a lot to do with who was sitting next to me and giving me that news, and having the support of people around me. You know, it's not me. Yeah, sure, I've got a part to play, but you know, they say it takes a community to raise a village. You know, when you're in that situation and you're facing those adversities, it's the community that pulls you through.
So you give credit to Diane and your family, your support network, but do you have that resilience naturally as a person, do you think, or is it something that's...
Look, I've always been a half glass full kind of guy. You know, I can't really speak for anyone else. And, you know, I think, you know, my dad, who's here tonight, has always really been somebody who thinks that you can do absolutely anything you put your mind to, and I'm proud enough to do the same thing. So I think attitude does make a big difference as well, and it's certainly, you know, that what's been given to me as I've been growing up has certainly helped me.
And Matthew's dad, Roy, is in the audience tonight. We're very pleased about that. So who would like to ask the first question? And then the second question in the second row here. Thank you.
Hello, my name's Nikki Blackwell. I'm an intensive care specialist at Prince Charles in Brisbane. I'm also a founder member of a non-governmental organisation called Alima, which works particularly in Africa, but also in Haiti, Baghdad, and other humanitarian crisis areas. So I have two things to say. First of all, a thank you to Matt. I heard your interview with your wife Diane on Radio National, and I think it should be used as compulsory teaching material for all intensive care specialists and trainees in conversations with family about moving from comfort care to comfort care from cure. It was quite extraordinary and a real revelationary moment for me to hear that conversation. Thank you both. And now a question to the panel, but particularly to Matt. The last time I heard Matt speak, he showed a slide of Sally Davies's, which showed the global problem of multi-drug resistance. And Africa is like a beacon out there. We haven't really talked about Africa yet. Having worked a great deal in Africa, where there are really no diagnostics whatsoever, and it's your best guess. You're very lucky now that we've got at least a rapid test for malaria, so now at least not every fever is malaria. And that's helped a little bit with resistance to malaria because maybe you use an antibiotic instead when it's negative. On the other hand, the NGOs have been present for 30 years plus now. We've been chucking out third generation cephalosporins and Cipro and the quinolones like lolly water.
So what is your...
my question is, what are we going to do about Africa? It's not India.
Okay, Matthew Cooper.
Yeah, sure. So we do something. I mean, in the past, I worked for a medical diagnostic company, and we deliberately launched our CD4 counter, which is for HIV, in Malawi, and the president of Malawi was involved in that launch. We had backing from the WHO and FIND, so I don't think it's acceptable just to say this technology is a Western technology, or we should try and trial it on African patients because it's easier or cheaper. What was surprising about the uptake and the story behind that was, although the technology seemed expensive, this is about $2,000, right? Which, you know, compared to the cost of care, it's 3 hours in ICU, was that it was subsidised. So the industry and the funding agencies such as the WHO said, well, this is a good thing to do. Let's subsidise the cost of the device and make sure that we just focused on getting it places. So you can see in Africa, mobile telephony has changed everything. They've flipped over lines, they've gone into mobile banking. In the same way that paradigm changes in health were driven by consumers for diabetes. Remember, diabetes was where we are now in the dark ages with a big machine in the 80s. You had to go to the hospital, get your glucose measured. A patient in Sudan would take a fingerprint test for glucose. There's no reason why we can't do the same. So I think it's a combination of really pushing the envelope. You know, this field is 300 years old. Let's really try and make those changes.
All right.
And whether we do it here or Africa, it's the same story.
Professor Lipman, you wanted to add to that?
Nikki, when I was listening to Matt Cooper talk, I was just thinking that we've got a place in society here with our sophisticated technology to be able to diagnose, to be able to set up the diagnostics better, to test the diagnostics better, to treat with dosing better, and to teach the world and to help the world solve some of the problems. So I was listening there and I thought, there's a lot in Queensland that we can do, not only for our patients, but we've got the technology, we've got the brains. I just do the work, he's got the brains. We can do it, and we're obliged to help. So that's my take on that.
And going on my Twitter feed, stories like Matthew Ames and the way he tells them just adds to that advocacy. So, yeah, I've been inundated with comments about Matthew rather than questions so far. But one question from Rebecca, thank you. Do superbugs need super drugs or actually just old ones repurposed?
It needs both. Some superbugs will be treated by old drugs that are being rekindled, phosphomycin, colistin, there's no question. And we're learning to use those drugs better. So we will be able to treat some of the superbugs with old drugs used more appropriately. However, there will be some bugs, the E. coli, Professor Shemri will talk about some of his E. coli, but there will be some bugs that need new drugs.
It buys us time. But fundamentally, we need new compounds to address these. We have extreme drug-resistant TB and bacteria. Now it's resistant to everything.
A tweet from Simona: how do we educate the public of the complexity of the issue when fewer young Australians are studying STEM subjects, being science, technology, engineering and maths? Professor Cooper, Dr. Young, who wants to take that?
I think it's a matter of improving health literacy in so many different ways. This is one issue that we've got to educate people about. We start at school, but then it's lifelong learning. So we just need to get out there and make things understandable. And I think what you've brought together here tonight does make those sorts of issues easier for people to grasp and understand and engage with.
So there is a technical problem, not educating the public, but you've got on the one hand a gentleman here that goes to the GP or goes to a department and is diagnosed as a flu-like illness and hasn't got a flu-like illness. More commonly, someone goes to the GP and has a flu-like illness and gets antibiotics unnecessarily. So unless you have a way of differentiating this problem, you're going to have a problem on the one hand, but a more common problem is the overuse of antibiotics for viral infections. Yeah. And I don't know how you're going to solve that.
That's an education for GPs, isn't it?
Well, it's a diagnostic problem as well as education problem.
All right, our next question from the audience, sir.
G'day. My question is about in the body, how many of these infections are actually caused by bacteria where antibiotic resistance is a fundamental property of the bacteria, other than a case where, for instance, the bacteria is in a biofilm and it's only resistant because it's in that certain resistant state? And are there other techniques that we can use sort of in combination with antibiotics? For instance, with bacteria and biofilms, once you treat it with a certain compound that disperses the biofilm, you can then treat it with normal antibiotics. So, yeah, I guess other than just making new antibiotics, what other sort of compounds should be investigated for non-antibacterial techniques which make things more susceptible to antibiotics?
Professor Cooper.
Sure. So that's a good suggestion. I mean, a lot of the time the bacteria is already resistant, particularly, I mean, Jess mentioned Southern Europe and Asia, it's a major problem. The other time, of course, is you get the wrong drug or a broad spectrum drug, and after the fourth drug, you've induced resistance, which Alexander Fleming talked about. So there are approaches to try and stimulate our immune system, because our immune system is what keeps us healthy most of the time. They're still early, they have some promise. The other way is to go to phages. So there's a virus which actually attacks a bacterium called the phage. And in Russia, in Georgia, they didn't have access to a modern pharmaceutical industry. And so they actually started to develop bacteriophage, they're viruses that attack bacteria. The problem with that is they're very, very specific. Nature is amazingly creative. And so there you kind of need a diagnostic because these things will only attack that particular bug. So longer term, we're looking at these enzymes that are specific for bacteria. It's like a broad spectrum antibiotic, like a grenade. It will cause collateral damage, toxicity, your normal gut flora, which is good, it will kill all of that. But you may have a couple of things left behind, whereas these phage are like a sniper rifle. They'll just go for the superbug. So in the centre, we haven't talked about it, but we have a couple of these crazy ideas that, 10% of the ideas should be really weird. And so we're doing a little bit of that in the background.
Professor Hoy's looking at you. the 10% of weird ideas.
I believe it.
Yeah,
IMB has a 10% blue sky policy.
You're saying think outside the square. Yeah. And go to our next question over here. Thank you, Professor.
Thanks for a very interesting discussion so far. I come from an emergency medicine background. The question with us type A type people is always how long until we can get this? So what's the timeline until things like the Manion are in the emergency department and we can actually use them?
Yeah, it's to be frank, it's funding driven. Clearly that's the key in this stasis for the Australian government to get behind this. For a new drug from where we are now, we'll be typically 7 to 8 years away from launch if things all went well through the clinical trials. Diagnostics, our world record was 10 months for a dengue diagnostic. This technology, if we had the appropriate funding, I'd say would be 4 to 5 years away from the market. But of course what we need to do, what we're already doing is saying to the clinicians, what do you want? What type of information do you want? Do you want it on your phone? Will Jeannette get upset if we bypass the Queensland Health IT system and put it on someone's phone?
She doesn't.
How will this all work? So there's a dialogue to have with clinicians and with the public. How do you want information? With glucose, we bypass that. The consumer goes to the chemist, buys a result, measures their glucose.
If you put it on the phone, I won't be able to report it so people can see it, which you want.
We can hook it all together.
All right, let's go to our next question. Thank you, ma'am.
Hi, this is a question for Dr. Young. So you were saying about the fact that pharmaceutical companies are obviously placing their funding elsewhere, obviously a huge issue. So did you know of any initiatives that are being taken to encourage pharmaceuticals to put their funding into antibiotic research more than they are at the moment?
No. NHMRC, of course, funds research, but it doesn't fund research in this space, because that's seen to be the pharmaceutical companies, but then we've got the work that's been done here by IMB. At the moment, I'm not aware of any decision by government to go in and actually then fund the development of new antimicrobials. But that's something we'll be talking about, I'm sure, when we get together in November, all the different stakeholders from across government and from across the different sectors, which will include pharmaceutical companies, I would think, to talk about these things.
From Glenn in our audience, in trying to come up with solutions to deal with superbugs, Matt Cooper, what are the risks and how likely are they that the situation will be made worse with superbugs?
Superbugs. Megabugs.
Megabugs.
Look, it's already happening in Greece and Turkey in particular, and in India and Pakistan. In our laboratory, we have 20 isolates that are resistant to all bacteria. So they're already here. They're not prevalent. They're not in our hospitals yet. But the fact that they're already here tells you, given those graphs we saw in the trend, if we don't do anything, in 10, 20 years, there'll be an issue. So it's not like the sky is falling, we're going to die tomorrow, but if we don't do anything, we're in trouble.
And for Professor Lipman on Twitter, a question from Stephanie. Why do you think it is that people still take antibiotics for granted, and what will it really take to change attitudes across the whole population to start using them responsibly?
Oh, can I pass that to Dr. Young? I think, while I was listening to Matt talk, there are two aspects. There's the now aspect that we need new drugs now, because we need to be able to treat the resistant organisms, and we need to be able to look at the new drugs, see how to dose them and how to use them properly. So that's relatively immediate, 4 or 5, within the next 5 years. At the same, so you can't, you need something new for the superbugs. at the same time, you need to teach everybody how to use them properly and not abuse them, which we're all learning. I mean, you know, I'm using a lot less antibiotics now than I did 5 years ago and 10 years ago. So we are learning, but just like we have to teach, everybody has to learn about this, we still need something now. So they're two, in my mind, when I was listening, they're two different aspects.
Okay, so I think we need to draw
I may not have answered the question on how you teach everybody, and I'll leave that to Jeannette.
I think we need to draw it to a close. But on that point, I would like to come to our panel finally and ask you this question. The glaring thing from where I sit is how you do this advocacy outside this room. How do you get people to outside this room helping to advocate for antibiotic resistance? And can I ask each of our panellists how you think that message can be spread effectively? And Dr. Jeannette Young, I might start with you.
Behavioural change is always very, very difficult, no matter what it is in health, and you just have to start with a burning platform. I think if you don't have that, then it's really hard to get a message out. I think we've got that here, and we've seen it, and you can start to see the media picking it up, you know, with multi-drug resistant TB and extremely drug resistant TB. So you're going back to having to do surgery to treat people with TB infections, and we're going to see more and more of that happen. So I think it's getting the message out in a whole multitude of ways to explain the issue to people.
Professor Lipman, you look at the amount of attention given to something like breast cancer research. How would you answer
I think
this?
my answer to your question is, I know this lady called Madonna. She's going to help us.
I think I have a phone number. So how do you think?
Well, I'm not sure I can answer that. I'm very restricted to intensive care. But I mean, when my wife goes out and her friends say, well, she went to the doctor and they didn't give me antibiotics, and I say, oh, that's great, and she says, no, they didn't like that. They want antibiotics. So it's public. I mean, it's doctor awareness, but it's also public awareness. You know, often people are going to a doctor to get something, and that's not right. I don't know how to solve it, but I know it's not right.
Such a terrific advocate, Matthew, and people listen to you. What would be your advice in terms of spreading this message?
Look, I've probably got a bit of a balanced view, because I certainly I didn't get the antibiotics, and I should have. So I think we need to be mindful to make sure it's a balanced conversation, that we don't go a little bit too far one way or the other. And for me, you know, coming from the patient's perspective, really listen to your body. I knew there was something really wrong. And I just really encourage people to just have a conversation with their doctors about viral versus bacterial, you know, and inform yourself and just understand. For me, you know, even before antibiotics, a simple blood test would have probably helped me earlier rather than later. So there are other ways as well, and explore those with your GP. You know, if you're not happy with the antibiotics, what are the other options? So I think at a public health perspective, having people armed with just the questions that they need to ask, you know, about viral versus bacterial infections, and you know, why do you think I've got a viral, why do you think I've got a bacterial, and why aren't I getting antibiotics? And I think that would go a long way to have those questions between patients and GPs.
Thank you. And Professor Matthew Cooper, last word?
3 things. Community, that's us, everyone in the room, it's everyone watching online. If we work together, we can solve things. Celebrity advocacy. So Michael J. Fox, you think of Parkinson's. Magic Johnson, HIV/AIDS. They made an enormous difference to funding, donations, research, really impacted disease. And then patient advocacy. So with cancer, there are huge advocacy groups. They're phenomenal. They do a lot of great work. And it's because there are many survivors. Unfortunately, with sepsis, half the people die. You know, Matt was right. He's lucky. He's a very lucky man to get through this. So community, celebrity advocacy, Daphne's fantastic. And then patient advocacy. And if we can work together as a community, we can solve the problem.
A great point to leave our discussion, I think. Ladies and gentlemen, a big round of applause for our panel. And remember, the key tonight is to spread that word, keep this conversation going, and that will ensure the work of the IMB Centre for Superbug Solutions and others is known. A safe trip home, and thank you.